set, प्यार.
Now, let us begin by taking a look at the definition of callus culture. A callus is an unorganized mass of plant cells that forms when a piece of plant tissue, known as an explant, is placed on a nutrient-rich medium with plant hormones like auxins and cytokinins. It is a common method in plant tissue culture, especially for regeneration and propagation purposes. Now, suspension culture is the next step, where it involves transferring callus-derived cells into a liquid medium. This culture are kept on a shaker to maintain a uniform suspension. This method allows faster cell growth and is ideal for scaling up biomass or metabolite production. Conceptually, both are in vitro techniques that rely on controlling the environment and growth regulators. While callus is grown on a gel-like solid medium, suspension cultures are maintained in liquid, offering better nutrient availability. While the main purposes or the main objectives of using these cultures are to first, to obtain genetically uniform cells for experiments or mass production. Second, to enhance the production of valuable secondary metabolites such as alkaloids. And third, to provide starting material for plant regeneration, genetic engineering, and protoplast isolation.
So for the next part, we'll be talking about the steps and techniques involved in inducing callus formation. As we know, callus is a mass of undifferentiated plant cells that forms when plant tissues are cultured in vitro. It's often used as the starting point for plant regeneration, genetic engineering, or secondary metabolite production.
The process begins by selecting a suitable explant which can be taken from various parts of a plant such as the leaf, stem, cotyledon, or root. This explant must be surface sterilized using ethanol or bleach to prevent contamination. After sterilization, the explants are cut into small pieces to increase surface area and improve the chances of callus induction. Next, the explant is placed on a nutrient medium such as Murashige and Scope or MS medium. This medium is enriched with macronutrients, micronutrients, vitamins, and a carbon source, and most importantly, the plant growth regulators.
So to promote callus formation, we generally use a higher concentration of auxins like 2,4-D or NAA, often in combination with a low amount of cytokinins such as the BAP or kinetin. The cultures are then incubated under controlled conditions, typically at 25 degrees Celsius, sometimes in the dark depending on the species. So over the next few days or 2 weeks, you can observe the formation of a soft, white, or yellowish mass of cells, which is the callus. Finally, callus is usually subcultured every few weeks to maintain its growth and vitality. So this technique is fundamental in plant tissue culture and acts as the base for further experiments or applications.
Assalamualaikum and hi everyone. Now I am going to talk about the establishment of suspension culture. So what is suspension culture? Suspension culture is a plant tissue culture technique involving the plant cells being grown in a liquid medium to encourage rapid cell division and biomass production. So now let us all take a look at the short explanation on the establishment of suspension culture. The process begins with the initiation step where a healthy piece of callus is transferred into a sterile liquid nutrient medium in a flask. This is followed by agitation using a rotary shaker with a speed of 100 to 150 rpm to keep the cells suspended, ensures even distributions of nutrients and oxygen, and prevents the cells from settling at the bottom of the flask. As the culture grows, regular maintenance such as medium renewal to supply fresh nutrients and provide that support. When the cultures become too dense, homogenization is carried out by filtering the culture through a sterile sieve to remove large clumps and return the smaller actively dividing cells. These filtered cells are then subcultured in the first medium, allowing the process to continue in cycles. Over time, the culture may develop into either cell-aggregates or dissociated cells as seen in the picture. Overall, suspension culture is a powerful method that enables large-scale propagation of plant cells under controlled and optimized conditions.
Hi everyone, I'm Anor and I'll be explaining the factor that influence the growth and development of the Callus and suspension culture. Okay, first is the most important factor is the plant growth regulator, where auxin is promotes cell elongation and root formation, while cytokinin stimulates shoot development. So the right ratio between this hormone determine whether we get the shoot, root, or just indifferentiated callus tissue. And the second one is environmental condition like light and temperature, and some cultures need a darkness to induce the callus, while other benefit from light. So optimal temperature usually between 23 to 27 help boost cell division and metabolic activity. Okay, for third, the nutrient medium composition. This include the macro and micronutrient, carbon source like sucrose and vitamin. So Morishike's cooked medium is widely used to support a rapid growth. So in suspension culture, a factor like agitation speed and cell density matter. Agitation help it distribute oxygen and nutrient evenly. But if the culture start to dense, cell might clump or compete for resources that's slowing down the growth.
So the application of Callus and suspension culture in plant biotechnology is first micropropagation. So Callus culture is used to mass produce identical plant from a next plant. And cell suspension allow faster multiplication because cells are dispersed and can grow rapidly in liquid media. Second is homoclonal variation. Callus culture can generate genetic variation useful for developing new traits in crops like disease resistance or stress tolerance. Third is genetic transformation. Callus tissues are ideal targets for introducing new genes. Transformed cell can regenerate into whole new plants carrying a desired trait, just like this corn. Fourth is germplasm conservation. Callus culture can be cryopreserved, like shown in the picture, for a long-term storage of rare or endangered plant species. And fifth is artificial seed production. Embryonic callus or somatic embryos produced in suspension can be encapsulated to create synthetic seeds like this. This is synthetic seeds. In conclusion, Callus and suspension culture are fundamental techniques in plant biotechnology, enabling the regeneration, genetic improvement, and mass production of valuable plant materials. Understanding their concepts, induction methods, and growth factors not only enhance tissue culture efficiency, but also supports the application ranging from micropropagation to genetic transformation and secondary metabolite production. Together, these tools play a vital role in sustainable agriculture, conservation, and the advancement of plant-based industry. That is all from us. Thank you. So the application of Callus
for the
and
next step.
suspension culture.
So